Welcome to this lesson on proteins, the remarkable amino acid chains that serve as the workhorses of biological systems. Proteins are among the most important molecules in all living organisms, performing countless essential tasks that keep cells and bodies functioning properly. Let's start by understanding what proteins actually are. In terms of structure, proteins are long chains made up of smaller units called amino acids that are linked together. Think of them like beads on a string, where each bead is an amino acid. As for their function, proteins perform virtually every task that needs to be done inside living cells. They are truly the molecular machines that make life possible. Now let's examine the building blocks themselves, amino acids. Every amino acid has the same basic structure. At the center is a carbon atom called the central carbon. Attached to this carbon are 4 different groups. First, there's an amino group, written as NH2. Second, there's a carboxyl group, written as COOH. Third, there's a simple hydrogen atom. And fourth, there's what we call an R group or side chain. This R group is what makes each amino acid unique. In nature, there are 20 different amino acids, each with its own distinctive R group that gives it special properties. Amino acids connect to form proteins through a special type of covalent bond called a peptide bond. Here's how it works. The carboxyl group of one amino acid reacts with the amino group of another amino acid. This reaction creates a bond between the carbon atom and the nitrogen atom, forming the peptide bond. During this process, a water molecule is released. This is why we call it a condensation reaction or dehydration synthesis. Once formed, these peptide bonds create the backbone of the protein chain. Proteins have 4 levels of structural organization. The primary structure, or first-degree structure, is simply the linear sequence of amino acids in the chain. The secondary structure, or second-degree structure, refers to local folding patterns that form within the chain, such as alpha helices and beta sheets. The tertiary structure, or third-degree structure, is the overall 3-dimensional shape of a single protein chain, formed by further folding. Finally, the quaternary structure, or 4th-degree structure, occurs when multiple protein chains come together to form a functional assembly. Each level of structure is critical for the protein's ultimate function. Proteins serve an incredible variety of functions in living systems. First, they act as catalysts in the form of enzymes, which speed up chemical reactions that would otherwise be too slow to sustain life. Second, they provide structure, like collagen, which gives strength to skin and bone. Third, they transport molecules, such as hemoglobin, which carries oxygen through your bloodstream. Fourth, they defend against disease as antibodies that fight off pathogens. Fifth, they serve as signaling molecules like hormones, with insulin being a prime example. And sixth, they enable movement, with proteins like actin and myosin contracting to make muscles work. Let's take a closer look at how enzymes work, since they're such important proteins. The process has 3 main steps. First, a substrate molecule, which is the substance the enzyme acts upon, binds to a special region on the enzyme called the active site. Second, once bound, the chemical reaction occurs, transforming the substrate. Third, the product of the reaction is released from the enzyme. Here's the key point. The enzyme itself remains unchanged throughout this process and can be reused over and over again to catalyze the same reaction with new substrate molecules. Several environmental factors can affect how well proteins function. Temperature is one major factor. Extreme heat can disrupt the protein's structure, leading to loss of function. The pH level, or the acidity or alkalinity of the environment, is another important factor. Changes in pH affect the ionic bonds within the protein and can alter the shape of the active site in enzymes. Salt concentration also matters because it influences electrostatic interactions that help stabilize the protein structure. Finally, many proteins require cofactors, which are helper molecules, either metal ions or small organic molecules that must be present for the protein to function properly. Denaturation is the process by which a protein loses its normal structure. It occurs when the bonds that maintain the protein's three-dimensional shape are disrupted by factors like extreme temperature, incorrect pH, or chemical agents. In the native state, a protein is properly folded and fully functional, able to carry out its biological role. In the denatured state, however, the protein becomes unfolded and loses its functionality. Think of cooking an egg. The clear egg white turns solid and opaque because the heat denatures the proteins, permanently changing their structure. Let's review the key concepts. First, proteins are chains of amino acids linked together by peptide bonds. Second, the structure of a protein determines its function, and this structure is organized across 4 levels from primary to quaternary. Third, proteins perform diverse and essential roles in living systems, including catalysis, providing structure, transport, defense, signaling, and enabling movement. Fourth, environmental factors such as temperature and pH can disrupt protein function through a process called denaturation. In summary, proteins are essential molecular machines that make life possible. Understanding how they're built and how they work is fundamental to understanding biochemistry and biology as a whole.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.